Hello everyone. The topic is cancer and cell therapy. So before explaining how cell therapy can cure cancer, first we will look out what is cancer and how it spreads in human body. So what is cancer? Cancer is a disease in which some of the body cells begin to divide without stopping and spread into surrounding tissues. So cancer can start almost everywhere in the human body, which is made up of trillions of cells. Normally, human cells grow and divide to form new cells as body needs them. When cells grow old or become damaged, they die and new cells take their place. But in case of cancer, cells become more and more abnormal. Old or damaged cells survive when they should die, and new cells form when they are not needed. These extra cells can divide without stopping and may form growth called tumors. These tumors are malignant, which means they can spread into or invade nearby tissues. In addition, as these tumors grow, some cancer cells can break off and travel to distant places in the body through the blood or the lymph system and form new tumors far from the original tumor. So, in this way, cancer can spread throughout the body. Now, what is the difference between cancer cells and normal cells? Cancer cells are less specialized than normal cells. That is, whereas normal cells mature into very distinct cell types with specific functions, cancer cells do not. This is one reason that unlike normal cells, cancer cells continue to divide without stopping. In addition, cancer cells are able to ignore signals that normally tell cells to stop dividing or that being in a process known as a programmed cell death or apoptosis, which the body uses to get rid of unneeded cells. So the cancer cells ignore these signals and they continue to divide without stopping. So here we can see in the first diagram that the cancer cells are dividing, and when they divide and grow, they kill the nearby healthy cells and form a tumor. So here we can see an animation of how CRISPR cure cancer. CRISPR is a new technique to cure cancer, and scientists are doing lot of research on it. So now we will see how CRISPR cure cancer. Here this is a mutated DNA. So what we will do is to break the DNA in two parts so that it loses its functionalities. So how can we do it? We can break the DNA wherever we want. So this is a Cas9 molecule which has the complement sequence of the bases when compared to the affected DNA which we want to divide. So this Cas9 molecule attaches with the affected DNA and breaks it into the two parts and at the specific places and damage the functionality of cancer cells. There are many gene therapies strategies which are implemented in patients, like changing the cells to increase or decrease its functionalities. There is a mutated protein called p53 protein which is present in nearly 50% of all the cancer cells. This protein alters the ability of cancer cells to kill themselves by a process called apoptosis. So in present days, the clinics are trying to insert healthy p53 gene into the affected cancer cells. They are doing this because mutated p53 protein interfere with the curing process like chemotherapy. The cancer cells become more insensitive to the chemotherapy, so by introducing healthy p53 protein enhances the effect of chemotherapy on cancer cells. Then these cancer suppressing genes are put into gene for giving signs in the cancer cells, due to which these cancer cells become more sensitive to the apoptosis and they will kill themselves. Many scientists are trying to introduce cytokines, which are immune stimulating proteins, so that the patient body can detect the affected cells in the body and destroy them. Another approach is to use RNA interference, which destroy immune suppressor proteins. Now we look at the part that how these tumors propagate to other parts of the body. Normally there is a forces between cells which bind them together, but in case of tumor, these cohesive forces become less and they lose cohesion and enters to other parts of our body through blood vessels and affect other healthy tissues. So a possible gene therapy is that target these genes and modify or alter them in such a way such that our immune system is able to recognize these cells and kill those cancer cells, so that these tumor cells won't be able to divide and propagate in other parts of our body. One approach here is that kill the cancer cells. There are some viruses discovered which can kill these cancer cells. So what we will do that inject those viruses in the tumor sites and allow them to grow there and kill the tumor cells. Also, there were some genes known as suicide genes which can help human body to kill these cancer cells. But all these therapies have many limitations because every cancer cells have different genome, so they require different gene to kill themselves. An alternative approach is to modify the immune system or to supercharge the immune system so that it itself go on and catch cancer cells and kill them. They should have the capability to find the cancer cells on their own. It is the fact that in a healthy person's body there are healthy cells which have the capability to find the precancerous cells and early cancer cells. But as time passes, cancer cells start coping themselves and start killing normal cells and become more danger that our immune system becomes unable to kill the cancer cells. These cancer cells harm the afferent and efferent arms of immune cells and immune system. So the approach is to remove the immune cells from human body by a process called apheresis. Then transfer these immune cells to a bioreactor platform where they were provided habitable liquid media which contain various nutrients, vitamins, cytokines and other growth factors. So by this process the cancer cells will become supercharged and then reinsert these immune cells to human body. Now every cancer cells have a phenotype associated with them. So we will modify the immune cells so that they fit in these phenotypes and kill the cancer cells. So until now we have discussed the way to cure cancer using cell therapy. If this is the solution to the deadly disease, then cancer is cured. But this is not the case. There are many limitations. So now we will see the limitations. So the first limitation is that the immune system could attack the modified cells. So as we have seen that we are modifying these cells. So when we inject these cells in our human body, then it can active our immune system and our immune system itself kill these modified cells. There are also chances of creating new cancer cells or triggering the existing ones, and we have to find a method to efficiently inject the modified cells into a human body. And there are also some ethical reasons that the government is not allowing the scientists to experiment with the living human beings.